Thank you, Mercy. And again, I welcome all of you to today's symposium. As Pfizer Symposium, we are focusing on the first 90 days of life. So this symposium will provide a comprehensive analysis of what we have learned from the past at present, especially since yesterday through this ISAD conference. We all had the numbers, we had the progress being made, and up to now, we all know that infant GBS disease in the first 90 days of life is now recognized as a global problem. This burden is here in Kenya, in Africa, and it's globally. We also know that existing standard of care for the prevention of infant GBS, like screening, IAP, where it exists, has limits and is not practical, especially LMSC setting. And it is also not sufficient to address the global problem. We've also learned that protective maternal GBS antibody is naturally transferred to the infant over the course of pregnancy. This naturally acquired maternal antibody can reduce risk for infant GBS disease, but may not always be in adequate amounts to provide the desired protection. So now we have maternal GBS vaccines which are in development and will take advantage of the transfer of protective maternal antibody. And through a maternal immunization program, we can potentially address all these issues. So today's symposium, we have 3 speakers who will help deepen our understanding of how this is going to be possible. We have Stephanie Schreig, who will present a recently published CDC study describing serological thresholds for reducing the risk of infant GBS disease through maternal antibody. Then we have Dr. Barbara Paul, who will provide an overview of the Pfizer maternal GBS clinical development program, including their global phase 3 Beatrix trial. And lastly, we'll have Dr. Gonzalo Perez, who will share lessons learned from another maternal immunization program, the respiratory syncytial virus, with a practical focus on the experiences of RSV maternal implementation in his country of Argentina. And I believe by the end of this engaging symposium, we will find that what we've learned more about and how our collective efforts are coming together as a community, and hopefully someday will help reduce the impact of this terrible disease. So as part of housekeeping, please make sure your phone is on silent mode. Those are the disclaimers for Pfizer. And without, we have conflict of interest from all the speakers. And without wasting much time, I'll introduce the first speaker, Stephanie Schreig. Stephanie, welcome.
Great. Yes, and thank you for the opportunity to present on this study. I'm showing here again the reference, so everything I say today and a lot more is actually in that paper. And I also want to remind everyone that Julia Rhodes, who was one of the lead authors on this study, is sitting right over there, and that's where you direct your questions after the symposium. And I want to remind everybody that I am no longer with CDC, though I was the PI on this study. And so I'm drawing only on publicly available information. And also to clarify, no Pfizer funds were used for the support of the study or for my support for this talk. We did have important co-funding for the study from the Gates Foundation, and then there was substantial government funding, and we also had a lot of contributions from the study institutions shown here. There was really an army behind the study, and I want to thank all of them up front. So the aim of the study was to provide U.S. evidence to inform an immunologic endpoint for a maternal group B strep vaccine. And just so that we're all understanding what we mean by immunologic endpoint, we heard a bit about this morning, and in Kirsty's talk in particular, we heard about how antibody against the capsular polysaccharide has been of interest as a marker for the conjugate vaccine products. And what we're talking about is, if you imagine this relationship between infant disease risk and antibody levels, is if you could describe this red curve really well, you would know how that relationship works. And it might not be exactly the shape shown here, this is just one example, but once you know the shape of that curve, you could pick any infant disease risk reduction of interest. The example here is an 80% reduction, and you can walk that over on the antibody concentration on the x-axis to see what concentration that corresponds to. And so when you hear people talk about serologic thresholds of risk reduction, it's that asterisk, that antibody value that corresponds with the reduction in disease risk. So our study was really aiming to describe this whole curve. And we did this by conducting a case control study that leveraged both in the US, we have a very strong state-based newborn screening program, and also this ABC surveillance you've now gotten to hear a lot about, but just a reminder, it's invasive group B strep disease surveillance in selected areas of the US. And just to describe the study a bit more, the cases came straight from that ABCs. The controls were enrolled in clinical centers in those catchment areas that were performing routine group B strep screening. The cases were all infants less than 90 days of age that had group B strep isolated from a normally sterile site. And the controls were infants born to a colonized mother, so they had some exposure to group B strep, but they did not go on to get invasive group B strep disease. When you hear me talk about serotype, the sample for the serotype in the cases was the disease-causing isolate, and in the controls, it was that maternal colonization sample serotype. And then in terms of where the cases came from, they came from 8 of our ABC sites, and from a long time period, because ABCs has been running now for more than 25 years, the cases came from 2010 to 2022. The controls came predominantly from 2 sites that got individual informed consent waivers, and the controls only came during the active running of the study, which was from 2018 to 2022. The primary sample for the antibody measurement in this study was the residual dried blood spot from this routine newborn screening. And the assay that we used to measure antibody concentration was the Pfizer Direct Luminex immunoassay. This assay was adopted by the Group B Strep Assay Standardization Consortium and then modified at CDC by Shanna Bolson, who's also here in the room, for use on the dried blood spot. And then we collected demographic and clinical information from medical records. Just to make sure that people understand the study, because there can be confusion, there was no vaccine used in the study. It's a natural history study. The controls had some opportunity to get group B strep disease. The primary exposure, if you're an epidemiologist in this room, is actually the infant anti-capsular polysaccharide IgG concentration at birth, and the primary outcome measure is whether or not the baby got invasive group B strep disease. And all of the results that I present will be stratified by serotype and by the standard definitions of disease onset. And the primary analysis method in this study, we felt that it was really important because it was an observational design to be able to adjust for covariates the way epidemiologists always do. And there was not an existing method that allowed for covariate adjustment. So one of my former colleagues, Nong Sheng at CDC, developed a method that we named the covariate adjusted logit model, or CALM method, to get the curve. And the real value of the method is that we could do the covariate adjustment. That method has been published now. I have the citation on the slide, and for people interested, that's where you can learn about it. Again, the method is to get this curve, so that was what we were trying to do with our data inputs. And we could only estimate curves for strata with at least 5 cases above the lower limit of antibody quantification, or the LLOQ. And I show you here the covariates that we included in the model. Some of these were our choice because we felt they could be confounders, and some were at FDA request. And then we did also perform some secondary modeling methods using methods that other groups have published, and you can read about those in the paper. So just some basic characteristics of the cases and controls enrolled in the study. The median delivery age for both cases and controls was around 30. There was very little maternal HIV in this population, and that's typical in the US. Just hopping to the intrapartum characteristics, I want to point out that we do have widespread IAP in the US, and so you can see that reflected here in both the cases and the controls. And then looking at the newborn characteristics, one thing that was important to us, particularly after conversations with regulators, was to try to have the controls reflect the gestational age distribution of the cases. And we were able to accomplish this for the 34 to 36 week preterm group, but we had a lot harder time getting controls in the less than 34 week group. So I just want to make that clear. Another thing that was really important is in this study, we were trying to get newborn blood as close to birth as possible. And so the timing of the dried blood spot collection, I'm showing you here on the left part of this slide. And the dark blue is blood collected in the first day of life, and the slightly lighter blue is 24 to 48 hours, and you can see that we had 88% in the 48 hours of birth, so close to birth. The other bar chart on the right is showing the timing of the dried blood spot collection relative to disease onset among the cases. So also it was important for regulators in particular that we're getting samples not well after disease onset, but at the time of disease onset. And you can see that we were very successful in that. The vast majority were collected before or on the same day as disease onset, and 96% in the first 3 days after onset. And this is just giving you a feel for the actual analysis. Sample size, we had to drop a few observations because of missing values for the covariates, and I'm showing you this by serotype and then by the control early onset and late onset disease status. And I've just highlighted in yellow here, just for your orientation, that serotype 1b early onset disease had the smallest sample size for early onset cases, and serotype 2 late onset was the strata that was the smallest sample size. And then just a very high level snapshot of the overall antibody concentration results. Again, I'm showing you here the results by serotype, and what you can see is the proportion of controls in the first column that had antibody concentrations above the LLOQ, that had some detectable antibody, and then I show you the same thing for early onset disease, and in the final column for late onset disease. And what you may notice is that more controls were above the LLOQ than cases. So we had very high proportion of controls above the LLOQ. Also, if you look at early compared to late, you'll see that there are more early onset disease cases that had antibody concentrations above the LLOQ compared to late onset cases. And then in that final column of late onset, the 2 values shown in red are just to highlight that we did not have 5 observations above the LLOQ, so we could not generate calm curves for those particular strata. And you can also see, if you look across the rows, that there is some serotype heterogeneity. So this is another raw data slide. This is just showing you the distribution of antibody concentrations in a more visual way. And, excuse me, the top part of this slide is showing you the early onset disease results by serotype. So each of these boxes is a serotype. And the bottom part of the slide is the same thing for late onset disease. These turquoise sort of boxes are for the cases, and the magenta boxes are for the controls. And just visually, you can see, looking across all of these plots, and we've seen some data like this also earlier today, that the cases have much lower antibody concentrations than the controls. And if you look over at the tables, the p-values for these are all quite highly statistically significant. I do want to point out that for early onset disease, serotypes 4 and 5, you can already see in the box plots that there's a little bit more overlap between cases and controls, and you will also see that the p-values are just at the marginal level. And then this is just a clue to you that you're going to get to see the serotype 3 bee swarm plots again, so prepare for that. So now I'm going to move into showing you the actual calm curves, and this is just reminding you what those curves are. And so we're going to get to see some real curves and not just hypothetical ones. And I'm going to show you the results for serotype 3 because it's the most common global infant group B strep disease serotype. So I'm just going to build out this slide. Again, here's that bee swarm plot you just saw showing the difference between cases and controls. And here is the actual sample size numbers that we had to work with in this light blue for early onset disease. So we had 21 cases that were above the LLOQ to generate the calm curve. And here is your first real calm curve for serotype 3 early onset disease. And then you can see where we chose to draw thresholds, and we did these at the 75%, 80%, and 90% risk reduction. And in calm, this is actually a relative risk reduction relative to the risk at the LLOQ. And we translate those into thresholds or micrograms per ml, and you can see that for early onset disease, serotype 3, these ranged from 0.27 at the 75% threshold to a value of 4 micrograms per ml at 0.9. So this slide is showing you similar layout, but for late onset disease. And again, you can see that the curve looks a little steeper than what we saw for early onset disease. The shape of the curve is steeper, and that translates into much lower thresholds for late onset disease, serotype 3, and so they range from 0.03 to 0.09 micrograms per ml. And I don't have time to walk you through each of the curves in detail, but this is just a summary slide showing the curves, with the top row showing the early onset disease curves, and then the bottom row is the late onset disease curves, again by serotype. And the main thing I want you to notice is that there are different curve shapes for the different serotypes, and also that some of the serotypes converged into step functions where the risk didn't really reduce until you hit a certain antibody concentration, and then it drops precipitously. And this slide is showing you the actual threshold values. I don't expect you to read all the nuances, but you can go to the paper if you want to know the actual micrograms per ml. The main thing to notice here is, again, you can see that visually that there are higher thresholds for early onset disease than for late onset disease. You can also see the difference between that the thresholds are generally lower for the 75% reduction than for the 90% reduction, and some serotypes actually look like they have quite high thresholds. Oops, see if it'll go. So we conducted quite a few sensitivity analyses. Again, this was an observational study, and even though we could put covariates in our model, we were worried about other sources of bias. And I don't have time to talk through these today, but again, they are all well described in the paper. And the bottom line for you to know today is that all of these fell within the 95% confidence bounds of the primary analysis. So we felt reassured after conducting all of these that we weren't sitting on some meaningful source of bias that we hadn't explored. So there are some real limitations to this study that I do want to touch on. The cases and controls don't arise strictly from the same population because our controls primarily came from 2 geographic regions. We did include geographic region in the CALM models to address that. And the cases also came from this wider span of years than the controls. And we did address this also, and you can see description in the paper. We were most concerned about whether blood spots that had been stored for a longer time might have had some degrading of the antibody, and we didn't see evidence of that. Additionally, the dried blood spot is the primary sample in this study, and it would have been ideal if we could have to collect cord blood. Still, we had a very high proportion, as I showed you, that were collected within the first 48 hours of life. So this was a good proxy for having a sample at the time of birth. We also had a limited sample size, particularly for some strata, and you saw that there were a couple of strata for late onset disease where we couldn't actually generate CALM curves. Nonetheless, this is the largest sample size to date for the sero-epi studies. And also, when we debated, like, could we run the study for an extra year or something to try to increase the sample size, I want to remind you that the cases actually came from a 12-year time period, and just running for one more year wasn't going to make that big of a difference, particularly for the minor serotypes, which were the ones that we wanted to boost in sample size. So it's hard to get large sizes for these kind of studies. And then finally, we did use a new analysis method, but this is now published and available, and we also applied other established methods. So just the summary, we were able to show, and consistent with other studies, that anti-capsular polysaccharide IgG antibody concentrations distinguished cases from controls, and we could show that IgG does seem to be a good proxy for disease risk, maybe an appropriate immunologic endpoint. We found that early and late onset curves didn't share a common threshold. And it also was apparent, as I showed you for serotype 3, that very low antibody concentrations at birth appear to confer late onset disease protection for most serotypes. The serotypes were also not all created equal, and there was heterogeneity in the thresholds, and the type 4 and 5 cases may be a place where if it would be possible to get any additional data, we could understand better why the cases and controls are more overlapping. And the covariates helped improve the curve fit, but they didn't dramatically change the threshold. I get to pass the baton to Dr. Barbara.
Jumbo. Just want to make sure you're all awake still. Okay. Hello. Good afternoon, everybody. Well, today I'm going to be talking to you, as soon as the slides come up, about the Group B Strep clinical development program at Pfizer. I am Barbara Pahud. I'm originally from Mexico. I've been at Pfizer now for 4 years. And I am the clinical development lead for the Group B Strep program and the clinical lead for the Beatrix phase 3 study that I am very excited to tell you all about. And while we get those slides up and running, I guess I could start by telling you the study has started. We started enrolling in August. You can check the countries that are up and running in clinicaltrials.gov. This is our vaccine, a 6-valent polysaccharide conjugate vaccine. As you already know, I'm not going to repeat what you've all heard here. The 6 most common serotypes are in our vaccine. These are conjugated to CRIM, which we've talked about as well. As you know, Pfizer has a very good experience with CRIM conjugate vaccines with our pneumococcal program. The dose level that was selected was 120 micrograms per half an ml, which is 20 micrograms per serotype without adjuvant. So that's our final dose, and I will explain to you how we got there throughout the presentation. And the proposed indication, of course, is to administer this vaccine to pregnant women to protect infants from all the diseases you've been hearing about today, including sepsis, pneumonia, meningitis, and of course against LOD and EOD. This is the GBS 6, because of the 6 serotypes, clinical development program. I'm going to start, as you can see here, with the boxes, telling you just one slide about each study. We don't have time to go through all of them in depth, but I have the publication in there so you can pull them and read them whenever you want. First, with our first in human, this study used the vaccine, the 6-valent conjugated with CRIM, and we studied different dose levels, 5, 10, and 20 micrograms per serotype, with and without alum. The vaccine induced robust serotype-specific IgG responses for all the serotypes, and as you can see here, antibody levels increased within 1 week. They peaked at 2 weeks, and they stabilized by 1 month, and they remained high out to 6 months, and you'll see some figures about this in following slides. This helps support further development for a maternal immunization program, and there's your reference in case you want to read more about this study. So moving on to our pregnant women study, that's our phase 2. It had 3 different stages. The first stage was non-pregnant, but our stage 2 and 3 were pregnant women. So the stage 2, as you can see here in the USA, UK, and South Africa, involved pregnant women where we repeated again the same different dose levels, 5, 10, and 20 micrograms per serotype with and without alum. And then we selected the dose of 20 micrograms without alum per serotype, so that's how we ended up with a 120 per half an ml, to move on to the stage 3 study. In short, this is the article that highlights the results of that study. The vaccine demonstrated a very acceptable safety profile, very comparable to other maternal vaccines. Infants born to moms vaccinated with this vaccine had higher anti-CPS IgG titers than those born to placebo. And at levels associated with what you've all been hearing about in this conference, levels associated with reduced risk of invasive GBS disease based on all these natural history studies that have been presented. The study described the IgG thresholds for each serotype associated with disease reduction of 75 to 95% in infants. And based on these safety and immunogenicity profiles, as I said, we selected the 20 microgram per serotype dose, and that's what moved on to further studies. As you can see in the last bullet, the IgG concentrations remained elevated throughout delivery, and the transplacental transfer ratios varied from serotype to serotype and with the different doses, but you can see the range for the final dose selected ranged between 0.55 and 0.74. Booster study. So here again is the publication in case you want to pull it, but what I want to highlight here is, remember our first in human study, you see that arrow. We brought those non-pregnant participants back 2 years later and gave them another dose of the vaccine just in case we decided, do we need to revaccinate in future pregnancies or not, and to start answering that question. So as you can see in the graphs with the different serotypes, and the lines are very similar to each other, you can see baseline placebo and baseline doses. Then when we vaccinate that first point, I don't have a pointer, I don't think, but you see the first is the bottom when we start, then vaccination post-dose 1, we increase the titers, as you know. Then 2 years later, that graph falls down a bit. So the levels remain higher than baseline, even after 2 years. So that is great news. It may be once we define what these thresholds are, that may be sufficient. You may not need another dose in the future, we don't know. That remains to be defined once we identify these thresholds. But then what's important about the study is you can see that it is boostable. That second dose 2 years later does increase the levels to higher levels even than what we had after 1 dose 2 years after the fact. So this just opens possibilities for whatever decisions are made once this vaccine hopefully is brought to licensure. Next is the phase 2B non-pregnant women study, where we co-administered with Tdap vaccine. And as you all probably know, pertussis tends to be a little bit finicky, so there was interference with pertussis, but we had adequate levels for tetanus, diphtheria, and the GBS6 vaccine. Pertussis does have lower immune responses when co-administered, as we see with other maternal vaccines, but also the clinical implications of this interference, as we know with other vaccines, remains to be determined. So now I come to the phase 3 study. And as has been adequately described, thank you, by others before me, even though we have a very small sample size in our study of 6,000, if you wanted to do a true vaccine efficacy study, you would need 50,000 to, I heard earlier today, 100,000 participants based on the variability of the serotype incidence in different regions and how many participants you would need in all the different regions. So for this reasons, with regulatory authorities, as you've heard, the decision to do an immunogenicity and safety study and then do a phase 4 post-licensure vaccine effectiveness study is what has been decided to move on with this study that we're doing. So here's our phase 3. The name of the study is Beatrix. In Pfizer, in case you don't know, we like to name our studies after either famous artists, like in the RSV study, or now we picked authors. So Beatrix Potter is who inspired us. And this is going to be including healthy pregnant participants less than 49, between 24 and 36 weeks gestational age, randomized 1-to-1 to either GBS vaccine, the 20 micrograms per serotype, without alum, or placebo. We are in 18 countries with adequate representation of high-income and low-middle-income countries. And as I previously stated, and you've heard today, safety and immunogenicity is what we're going to be looking at, not vaccine efficacy. Here are the key maternal inclusion-exclusion criteria. I'm not going to read all of them out to you, and there are more than what's currently listed here, but I just thought you might want to see them. In general, healthy, less than 49-year-old women between 24 and 36 weeks gestational age. We have early dating ultrasounds that need to be performed in the first or early second trimester, and they need to be able to give informed consent, deliver at a hospital. We're not allowing moms who intend to deliver at home. And important exclusion criteria is if they have a current history in the current pregnancy of GBS, then they cannot participate. But if a child had GBS, if a prior pregnancy had GBS, that mom can participate. And of course, previous vaccination with any GBS product would exclude them from participating. Here are primary safety endpoints, and it is to describe the safety and tolerability of our vaccine when administered to healthy pregnant women. We are collecting, of course, for the first 7 days post-vaccination, local and systemic reactogenicity, for the first month post-vaccination, adverse events, and throughout the time that the moms stay in the study, medically attended adverse events and serious adverse events. For the babies, from the moment they're born until 1 month of age, adverse events, medically attended adverse events for the first 6 months of life, and serious adverse events throughout the study. For our primary immunogenicity endpoints, you have heard throughout this conference the importance and the diversity of levels necessary for each serotype. So you will see that we are looking to assess GBS-induced IgG antibody levels predicted to protect against invasive GBS, LOD, and EOD by the individual serotypes. So we have an individual immunogenicity endpoint. In addition, we have a different immunogenicity input looking at the aggregate predicted vaccine effectiveness, combining all 6 serotypes, both for LOD and EOD. Letting that sink in. In addition, our other secondary immunogenicity endpoints include, as has been discussed, OPA antibody titers. And so we are going to assess the ability of our vaccine to induce OPA titers at birth in infant participants born to moms that were vaccinated with a vaccine. And because, as I mentioned, our vaccine is conjugated to CRIM, we are also describing the IgG responses to active immunization with diphtheria, toxoid-containing vaccines, because CRIM is a derivative of diphtheria, so we are looking to see there's interference there, and PCV, because PCV is also conjugated with CRIM. So there is a subset of participants in our study that are going to be receiving all their vaccines per their normal childhood immunization schedule, and these assessments are going to be done throughout the first year of life. I'll show you one example of what that schedule looks like later. So this is our schedule of activities for the moms. You can see they have a screening visit zero. Not all countries do that at separate days, but for example, in the United States, visit 0 and 1 are the same day. But it may be that in other countries we need to bring the mom in ahead of time to get some of the criteria determined. Then we have, of course, blood draws. As you can see, if visit 0 and 1 are combined, it would be just wine. Otherwise, it might have blood drawn at visit 0 to determine if they are HIV negative, hepatitis B negatives, etc. Visit 1, pre-vaccination blood draw. Vaccination occurs on day 1. Then we follow them, as you can see there, for 7 days, e-diary for the systemic and others reactogenicity. 1 month for adverse event collection, the medically attended adverse events, and adverse events of special interest. So adverse events of special interest are adverse events that are systematically collected because we want to make sure that they are reported regardless of the principal investigator's opinion of whether they're serious or not, reportable or not. So adverse events of special interest for us, for the mom, for example, are preterm delivery, hypertensive disorders of pregnancy, and preterm labor. Preterm labor being a mom who goes into preterm labor but delivers at term, versus preterm delivery, they do deliver preterm babies. And then finally, the SAE collection throughout the 6 months. For the infants that are part of the regular study, cord blood collected at birth, adverse event collection for 1 month, medically attended adverse events, serious adverse events, and the adverse events of special interest for the baby, preterm birth, low birth weight, group B strep disease, which we don't expect to find many, but if we find them, we want to know about it, and developmental delay. Now, this is an example of one of our countries, Argentina, and how the infant subset looks. Basically, the same schedule as the infants that I showed you, plus all the infant vaccines required by that country will be administered, and blood collected post-priming doses of diphtheria, tetanus, pertussis-containing vaccine, and pneumococcus, and post-booster. So that's why you see so much blood draws, because these countries have different schedules than the United States, than Africa. So each country has a different looking schedule mirroring their own requirements. This will also provide good information about the differences of responses in different countries, of course. Now, as you know, once this vaccine hopefully is licensed, it needs to be implemented and needs to be recommended for us to be able to do a phase 4 vaccine study. You have heard a lot about the potential design of this study and that it will be, of course, designed based on where the vaccine gets approved. It will require, again, recommendations, sufficient uptake, adequate GVS surveillance in infants, all things we've been discussing throughout the day, and the ability to link maternal vaccination status to the infants. Various methodological approaches have been discussed today, so I will skip those. And challenges, of course, is where do we have IAP? How is it implemented? How will we study that in these phase 4 studies, et cetera, and variation across countries? In conclusion, clinical trials have demonstrated an acceptable safety profile for a vaccine, with most adverse events being mild or moderate, similar to any other vaccine administered during pregnancy. The 20 microgram per dose without ALPO per serotype, so 120 total, without adjuvants, was selected for further development. This vaccine generates good antibodies that are transferred to infants successfully, reaching levels in the infants, which is, again, at the end of the day, what we care about, that are associated with reduced risk of invasive GVS disease. A booster dose when administered was well tolerated and elicited a very good serotype-specific immune response that was consistently higher than those observed after the first dose. Phase 3 trials have started, and we are working together with regulatory agencies and all of you in designing these phase 4 studies and planning our next steps. With that, I would like to pass it on to Gonzalo Perez-Mark, one of our investigators for this trial in Argentina, and Pascal Rivas, which is, of course, next to talk to you about his experience with the RSV vaccine and how that affects the GVS program. Thank you, Gonzalo.
So, hello everyone. Thank you very much for the invitation to this conference. It's really an honor to be part of this conference, surrounded by such distinguished colleagues and friends here. My name is Gonzalo Perez Marc. Like Barbara said, I'm a pediatrician, researcher, many years dedicated to clinical research, particularly in the fields of respiratory viruses and viral infections. And today, building on this experience in my country, Argentina, with the maternal RSV vaccine, you know RSV is a respiratory virus that often causes lower respiratory tract infections in newborns and infants. I presented a talk that examines how maternal RSV immunization policies translate into real-world evidence and uptake and program success. So I will focus in comparative perspective across countries, and implementation, how we make it work in practice across diverse settings and countries and regions and populations, and actionable lessons. So to understand a little bit the implementation success, we must first examine the key factors that we think were the principal reason that why we achieve this successful program. No? First, we will see that uptake is not just a number. It reflects policy, access, and the effectiveness of communication. I will come again and again with these three pillars, policy, access, and communication. Second, we will ask what vaccine uptake rate tell us about the implementation of our program by comparing experiences across 3 different countries, the UK, Uruguay, and Australia. After that comes Argentina. In the surprise, we will focus on our experience in Argentina, offering practical insights from the implementation of the RSV maternal immunization program. If we see, I already told you that a successful maternal immunization program requires multi-layer framework. You can see here some of the factors that are working in this kind of program: the policy, the vaccine recommendation by the government, data on disease burden and vaccine safety, vaccine costs, political will, community awareness, confidence. So many factors, but today's presentation will focus on 3 independent pillars that I already mentioned, that in my view, they underpin every successful maternal immunization program: a clear national policy, operational access, and effective communication with the different stakeholders. I believe that the program can only succeed when these 3 pillars really work together. So let us move to the implementation. And you can see here in the next slide that in terms of policies and access, for example, we can find a substantial heterogeneity worldwide in reimbursement systems for pregnant individuals, different structures depending on each country. And you can see here some of the examples from the United States, Argentina, Brazil, France, Finland, Australia, and also in RSV vaccination windows, starting from the 24th week of gestational age, like, for example, in countries like Finland or Poland, from the 28th week, like Brazil, Australia, South Africa, or between 32 and 36 weeks, like in Argentina or United States or Uruguay, and also in seasonal strategies. RSV seasonal versus year-round administration. You can see here, for example, in Argentina, we have seasonal administration like in the States. In Brazil, you have all year round, like in Australia and in England. So here are the examples of RSV maternal vaccine uptake rates across several national immunization programs. This is a comparison of vaccination implementation across the UK, Uruguay, and Australia. You can see how UK vaccinates from 28 weeks of gestation, year-round, and free of charge for every pregnant mother. The same in Uruguay, free of charge, but seasonal administration, and 32 to 36 weeks. And in Australia, again, 28 year-round and funded for all pregnant women. And all of these countries, they achieved coverages over 50%. You can see England, for example, 57.8 or 71 in 2025 season. In Uruguay, they implemented the program during 2024, but only in the last 2 months, August and September, following Argentina. And in Australia, 66% of the coverage in 2025. In terms of access, here are the results of the Bronx Stop study, which is continuing throughout this season, and they're informing new results. This study reflects maternal views on RSV vaccination during the first season of implementation in England and Scotland. At the top of the slide, we can see that most mothers, 86%, agreed or strongly agreed that the RSV vaccine was necessary. But you can see that, however, at the bottom of the slide, the results show that logistical barriers were an important factor for unvaccinated mothers. Despite the desire to receive it, 65% of the unvaccinated mothers never disagree with the vaccination, for example. You can see that even in high-income countries, there are some barriers for the access. In that regard, it seems that comprehensive data on the safety and effectiveness of the maternal RSV vaccine may help to reinforce public confidence and improve coverage rates. And this is the Argentinian experience with the vaccine. In my country, in Argentina, it was the first country in the world to have an RSV maternal immunization program, the vaccine inside the national program. And this is a summary of the key characteristics of my country and its health and vaccination system. Argentina has a population of 46 million and is a federal country with 24 provinces, each with its own health system. But our national healthcare system is composed by 3 insurance subsystems, including only public, nearly 40%, and social security and private insurance, nearly 60%. And we have a national vaccination law that defines vaccination as an individual right and social responsibility. Our public system is free for everyone that lives in the country. The national vaccination law also establishes free and universal access to vaccines, ensures equitable distribution across the country, and when it's supervised, and the recommendations come from an ITAG and a National Vaccine Safety Committee. The Ministry of Health has the role of leading in health policy and coordination, including procurement, distribution, and monitoring of the vaccines. So we discussed the importance of policy. This is the pathway we follow up to the launch of our RSV vaccination program. From 2014 to 2023, we only had available the palivizumab for the highest risk infants. And after they published the results of the MATIS trial, the phase 3 efficacy trial, in middle 2023, in September, our regulatory agency and MAT approved the RSV pre-F vaccine for pregnant women, like I said, between 32 and 36.6 weeks. In November of 2023, our NITAG recommended the seasonal combined prevention strategy, and very fast in December, the Ministry of Health included RSV pre-F in the national immunization schedule. The recommendation for the NITAG was combined to use the monoclonal antibodies for these newborns whose mother were not vaccinated, but the primary strategy was the maternal vaccination. So very, very fast, again, in 2024, like 6 months after they published the results, we started with the first national season RSV pre-F vaccination campaign that went from March 1st to August 31, because our RSV season is during these months, usually a typical RSV season. Well, after the first year of the program, we had, I think, good news. In the first year, Argentina achieved coverage rates for RSV maternal similar to these maternal vaccines for influenza and DTaP, which have been used for over 10 years in our country. And you can see that over 147,000 doses were administered, with an overall coverage of 67.8% in the first year of the cohort of the pregnant mother. Each jurisdiction, like I said, is a very federal country, carry out his own communications campaigns tailored to their specific settings. However, we found that coverage, as you can see, varied widely by region, with some provinces close to 90%, like Tierra del Fuego, in the south, and Mendoza, for example, and others below 53%, like Formosa and Misiones. So the provinces with a higher coverage were those that implemented more aggressive and sustained communication campaigns targeting both healthcare professionals and the general population. But we know that coverage alone is not enough. Usually we need effectiveness and data to validate policy. And after the first season, several research groups conducted in Argentina multiple effectiveness and study and impact studies. And here you can see the main results for them. Our study, the Bernie study, the third one, that was a collaborative study with Pfizer. And to maintain the tradition of the names, Bernie is a very, very well-known artist from Argentina, in this case. But many other studies found as a primary outcome with the RSV hospitalization or severe LRTI, an effectiveness of, you can see, 78.6% to prevent RSV hospitalization up to 3 months, and up to 6 months of 71.3%. So, and you can see similar results in all the studies that were done in Argentina. Based on these results, we can state that the real-world evidence reinforced the success of our program implementation, and that of course, the effectiveness was consistent with the data that the phase 3 trial showed. And after 2 complete seasons, no safety signals from our surveillance system were reported in any moment. With this evidence, Argentina moved into the second year. The last season, our season, was in 2025. Again, between March and August, and the Ministry of Health extended the vaccination season from 6 to nearly 10 months because of these results. In parallel, the Vaccinology Society, like I show you here, focused on communicating the results. We already said, like policy, access, and communication. For example, here are some examples. They held virtual meetings focused on providing information and tools for healthcare teams regarding RSV vaccination during pregnancy, discussion with field experts on implementation, the frequent asked questions of the physicians, useful resources, province-level experience, discussions in which professionals with experience in pediatrics, in obstetrics, in vaccination, and vaccination presented real clinical scenarios that allowed for reflection on practical aspects and current recommendations. So many of these actions were led by our Ministry of Health, and in some cases of the Ministries of Health, or Secretary of Health of each province and jurisdiction. In this way, in the second year, Argentina multiple departments achieved 100% of coverage. And like I said, an extended seasonal campaign was implemented starting from January 2025. You can see here in the first map is a summary of what we already saw in the other slides. Is the coverage of 67.8% in the first season. You can see some of the jurisdictions and provinces that are in red are under 70% of coverage, and in green you have some in the most southwest in Ushuaia, in Tierra del Fuego and the capital city is Ushuaia, between 90 and 100%. And in the second map and the third map, you can see the second season. The second season where all the provinces beside Tierra del Fuego were under 86% of coverage, with a total coverage for the country of nearly 65%, with over 184,000 doses administered. And you can see in the third map, you have some of the departments in blue are the ones that achieve this 100% of coverage during the second season. So which are our next steps for the program? Of course, assess the feasibility of offering vaccination year-round, extend the vaccination window to 28 weeks of gestation or 24, and make it easier for OBGYNs to co-administer it with other vaccines like Tdap, like we have in Argentina for pregnant women. We have Tdap, flu, and COVID, and why not in the future with a new GBS vaccine. In parallel, the revaccination in the event of a new pregnancy was recommended in Argentina. So up to this point, we can see that a clear evidence-based policy is the foundation of successful implementation. Real-world evidence allows us to address the key questions raised by the public, the healthcare professionals, and decision makers. Here, for example, define the vaccination window, when is it best to give during a pregnancy, what outcomes are you aiming to prevent, how much time do you need to transfer antibodies, safety, efficacy, effectiveness, data supportive to the window, achieve clarity on the seasonal strategy, seasonal, year-round, duration of protection, epidemiology of the disease, providing clear guidance on co-administration. Can you give it with other maternal immunization or not? That we are studying right now with Bernie study, for example, and develop a revaccination policy for subsequent pregnancies, like I said, we did in Argentina. Can you give it again in the next pregnancy? Do you need to in your country, in your region? So in conclusion, a successful maternal immunization program, every program of this kind require aligned policy, access, and communication with a real-world validation. Yeah, the program starts with a policy that supports access and implementation. Communication at all stakeholders level, like I said, is key from policy to provider to patients. The real-world evidence, the importance of the phase 4 studies, like Barbara already said, effectiveness and safety allows all the stakeholders to see the policy and the program implementation is working and can inform potential updates to the policy, like happened in Argentina during our second season, and learn from each other. See what works and does not work in different settings, use the learnings like this one to inform evolving policy, access, and communication. So thank you very much, and now we are having a panel discussion.
Thank you to all the speakers. That was great. And to the audience, can we appreciate our 3 speakers for today? So we are going to switch gear to the panel discussion, and for the interest of time, I will not take questions from the audience. I will just ask the speakers, and if you have more questions, you can look for them during poster session. So my first question will go to Dr. Barbara. So in your presentation, we saw you are in the phase 3 trial, there was monitoring of safety events. So can you elaborate further how safety data was monitored during the Beatrix trial?
Yes. So safety data is collected, as I mentioned, 7 days post-vaccination, 1 month following vaccination for adverse events, and throughout the time of the study, serious adverse events and adverse events of special interest. In addition, as you all probably know, many of you are investigators here, so you know this better. And please come find me if you're an investigator. I would love to meet you. I forgot to say that earlier. We also have a data monitoring committee that is independent of Pfizer that meets at least quarterly, more often if need be, to review the data unblinded. We don't get to see, of course, the unblinded data, but they do. So that's how other studies have been halted whenever there's a safety signal. So we want to, first and foremost, to put the safety of our participants ahead of it. So we have many medical monitors that review the data daily, and then all this aggregate data is presented to the Data Safety Monitoring Committee. And I'm happy to answer any of their safety questions if people want to find me later.
Thank you, Barbara. The next question goes to Stephanie. In your thresholds, antibody thresholds, we saw the late onset disease appear lower than the early onset. Do you have any hypothesis how that is?
Yeah, thanks. I mean, we have hypothesis, we don't have certainty. But this trend has actually been seen now in a few of the sero-epi studies, not only in ours. And we think that it most likely comes from the epidemiology differences between early and late onset disease, where the baby probably gets a much bigger exposure of bacteria in early onset disease, both through passage of the birth canal and also maybe the ascending infection. Whereas for late onset disease, it might be a more limited dose that the baby gets exposed to, and so a smaller amount of antibody might be needed to take care of that.
Yeah, thank you so much. Our last question goes to Gonzalez. I happen to be doing a lot of RSV work, and at this point I would like to know that you have experience in introducing and implementing the maternal RSV vaccine. So what do you think will be the most important factors for the successful implementation of GBS maternal vaccine?
Okay. Yeah, that's a good one. Considering that many countries, or most of the countries, don't perceive the GBS disease or GBS sepsis as a major public health problem, I believe the best way is to maintain attached to these pillars that I told you, like policy, access, and communication. And I mean, when I mean policy, I mean to make the burden of the disease, first of all, visible, not only for the public, but also for, that happened with RSV in Argentina, with OBGYNs, for example, for all kind of healthcare workers, especially for those who are involved in treating patients, in mothers and newborns and infants. I mean, OBGYNs, pediatricians, nurses, midwives, social workers, and not only in burden of disease, but also in efficacy, effectiveness, safety, and when I mean to take actions in terms of access, I mean to make the vaccination like effortless during the antenatal care. That helps a lot when for example, the government in Argentina is leading the implementation of the building and the implementation of the national program. If you can add your vaccine to the national program, that helps, of course, a lot. And if not, you can start with the vaccination in regions or parts of the country where the vaccine acceptance is higher. And of course, getting new real-world data, because the healthcare workers need it. I was telling you that the OBGYNs, of course, need to have this data for the co-administration and to have broader windows, and communicating everything that we said, and focusing in the specific groups. OBGYNs, they always want to know about safety. Is there any adverse event for the mother, for the baby, and not only co-administration and vaccine windows, but pediatricians, for example, are more focused in efficacy and effectiveness, because they know that vaccines are safe and they work the whole time. And sometimes you have, for example, midwives or nurses or social workers asking about supply logistics. So I will focus for this in again, this policy, access, and communication. Use all these strategies, and I think that could guarantee to have a successful vaccination program.
Thank you so much. So for the audience, those are so many takeaways, and that brings us to the end of this session. Thank you, everyone, for your time. Thank you.
Thank you.
I hand over to Mercy.
Thank you. Thank you very much, Joyce. Let's appreciate the Pfizer team, and let's appreciate the speakers for the symposium. You can take your seats. Thank you very much. Thank you for sharing your knowledge and all your presentations. You can take your seats.